very much decreased transcription, whereas euchromatin transcriptionally accurate, or at least potentially so, because it's not condensed. And so again, you have this idea that different cell types can have different regions of their genome that is accessible to these different transcription factors. So you have all these levels of control allowing different cells to be able to express different genes, allowing them to make different proteins, and therefore have different functions. Yes?
During the embryo, the initial concentration, is it set in the embryo or while, because the cells are dividing, right? These are totipotent or whatever. They're stem cells, they're dividing. Is the gradient that they go into these 3 layers already preset by the mothers?
So you're talking about how do you get mesoderm, ectoderm,
yes,
mesoderm?
Yes.
We're going to talk about that in just a minute.
Oh,
So, no, it's fine. I mean, it's a perfect question, because really what it comes down to is, okay, I really just told you that, okay, different cell types transcribe different genes, but I haven't told you how those initial differences get set up. And that's really what the question is. How do those initial differences, how do you go from one egg that's fertilized to multiple different cell types that have different transcription factors being active? We're going to talk about that in just a bit. But before we go into that, I want to talk about how it is that scientists actually study transcription on a whole genome level. So are there any other questions about transcriptional control and transcriptional differences between different cell types? All right, so there are 2 basic ways in which, well actually there are 3 basic ways in which researchers can take a look at the expression of genes in cells. The oldest, by using northern blots, this I'm not going to really talk all that much about, but basically what you do is you isolate RNA from cells. You do electrophoresis, which is where you separate based on size. You then transfer that RNA onto a membrane. And then you probe that membrane for whatever gene you're looking for. And the way that you do that is you make a single-stranded version of your gene that can complement the RNA. So your probe is a labeled single-stranded version of the gene which complements the RNA. And so then what you're able to do is you're able to, for example, take RNA from a nerve cell, a muscle cell, a liver cell, an epithelial cell, and see whether or not a specific gene is expressed in those different cell types. And so this really only works for one gene at a time. And so again, if you're studying a particular gene and you want to know what cell type it's expressed in, this is the way to go about doing it. However, this doesn't really tell you anything about the overall transcriptional profile of any one specific cell type. So it doesn't really tell you what is different in transcription between a nerve cell and a muscle cell. And so this is a very, very, like I said, it's an old method, and it's very limited in what you can find out. Yes?
How do you prevent the DNA from co-purifying with the RNA?
How do you keep the DNA from co-purifying with the RNA? Well, what is it that mRNA has that DNA does not have?
The A tail.
Poly A tail, right. So you can use that poly A tail to purify away the RNA from the DNA. So what you can do actually is, what they do is they make these agarose beads that have poly T attached to them. So the poly T tails bind to the poly A on the mRNA, and then all you do basically is you lyse open the cell, lyse open the nucleus, so it releases all the RNA, all the DNA, incubate them with the beads, pellet it down by centrifugation. Your DNA and all your other non-mRNA RNAs stay in the supernatant. Your poly A RNA comes down. Very easy to purify. Any other questions? All right, so then if you are looking more at the whole transcriptome of a specific gene, a specific cell type, the best, or one of the best ways to go about doing that is by using something called microarray technology. And what's great about microarray technology is it allows you to compare whole genome transcription between two different cell cycles. So, this could be comparing two different cell types. It could be comparing cells, the same cell type, under different conditions. So for example, you might want to take a look at the expression differences of pancreatic cells in the presence or in the absence of glucose. So the same cell type, different conditions. Or you might want to take a look at cells during a developmental time course. So you're looking at a group of cells at different points during development to see how overall transcription changes as development occurs. And so again, what it really all comes down to is comparing the transcriptional profile of different cells under different conditions. Any questions about that? About what this technology is used for? All right, so then how does this actually work? How do you actually do this? So again, this whole idea is that microarrays can compare the expression of two different cell types. And so what you want to do is, first of all, you have to make your microarray chip. And the way you do that is you collect a bunch of gene-specific DNA, you amplify that by PCR, and then you print that onto a glass slide. So what that means is that you're going to put a spot on a glass slide for each gene that you're interested in looking at. So if your organism has 40,000 genes, you're going to have 40,000 spots. Because you're going to want to take a look at where each spot represents each individual gene. So then once you've got that, what you need to do then is you need to get your RNA samples from your cell types. So in this example, let's say you are looking at the difference of transcription between a nerve cell and a muscle cell. And so what you do is you collect RNA from the nerve cell, and you label that RNA with a fluorophore that fluoresces red. You take your RNA from your muscle cell, label that with a fluorophore that fluoresces green, You mix them all together, you hybridize them to the glass plate, so now each of these individual RNA molecules is going to find the gene that is homologous to the gene that it can complement, and it's going to stick to that spot on the glass plate. Everyone follow me so far? Any questions about that? So again, all we're looking at right now is whether or not these individual RNA molecules are able to hybridize to their gene. Okay? You wash it, so you wash away all the unbound RNA, and then what you do is you scan the chip, you scan the slide for red fluorescence or green fluorescence. Red fluorescence means that this gene that was plotted here is expressed in nerve cells because it's fluorescing red, because it has RNA from the nerve cells. This spot here, fluorescing green, which means that this gene is expressed in muscle cells because it is bound to RNA that was derived from muscle cells. This spot here, which is black, means that no RNA bound to it. So it means that this gene is not expressed in either muscle or nerve cells. And the spot that's yellow, you get that when both RNA from the nerve cell and from the muscle cell bind to the gene. So this gene is active in both cell types. And so by looking at this array, you can see which genes are transcribed only in nerve cells, which genes are transcribed only in muscle cells, and which genes are transcribed in both cells, and which genes are transcribed in neither cell. Any questions about that? And so again, this is only one small section of a gene array, which again can be tens of thousands of genes. Any questions about that? So then how do you go about analyzing this? Well, again, this is just to give you an idea of how large these arrays are. They're tiny. So you can see this chip you can see this glass slide right here being held by a person, and you can actually see the little individual spots here. So this pretty much probably has something like 4,000 or 5,000 genes on it. So it's really, really easy to do large genome analyses in very, very small amounts. And so what information you get out of this is, again, you take a look at how genes are expressed. So what you want to do is take a look at what genes are coordinatedly expressed. So in this particular example, we have each row represents a specific gene expression. So this row represents expression of the ETS2 gene. Okay. Across the top here, so each column represents a different cell sample. This represents expression, for example, of a different transcription factor. And so all of these cells are transcribing this transcription factor, H1P, and you can see that H1P activates all these different genes. So all these genes are activated by H1P. Compared to, for example, SOX, a different transcription factor, which turns off a bunch of genes, it turns on others. So again, it's the idea that different transcription factors are programmed by the way they can bind to DNA to activate or deactivate different sets of genes. Oftentimes, those sets of genes are all involved in a very similar process. So for example, in this case, we're looking at is transcription after cells have been given glucose. And what ends up happening is that you can see that there are certain sets of genes that get turned on late in the time course. So if you give the cells glucose at time equals 0, and then take samples at different time points, up to 24 hours, and look at the expression of different genes, you can see that there's a bunch of genes that all cluster, that they all get turned on at about 4 hours of development through 24 hours. And it ends up that all these genes are all involved in cholesterol biosynthesis. So it's again this idea that genes that are involved in the same process are all being regulated in the same manner. Just like the situation right here, these